Okay, thank you everyone for coming. Kavya, would you be able to take a screenshot of the participants list while I start us off just for attendance purposes? Thank you. I forgot to do it last time. Okay, so welcome everyone to our second workshop of the year, Designing Procedures and Online Sources of Data, and it's presented by the gsdsef Outreach Committee, which is a student-led group that is volunteering to organize these workshops. So first, let's do introductions. I'm Andrew. I'm a senior at Canyon Crest Academy. This is my third year doing the science fair, and I am interested more in the bioinformatics and biotechnology aspects of science. And one fun fact about me is I have 4 younger siblings. Kavya, you can go next.
Yeah, so hi, I'm Kavya. I'm a junior at Westview High School, and my project category is computational biology, and this is also my third year doing science fair. Fun fact about me is that I have a pet parakeet.
Oh my, wow. That's cool. Eleanor?
Hi guys, my name is Eleanor. I'm a junior at Mount Carmel High School. This is going to be my third year in science fair, and my project category was bioinformatics. A fun fact about me, I like drawing.
Spencer?
I'm Spencer. This is my second year doing science fair. My previous project was in behavioral and social sciences, specifically cognitive science. A fun fact about me is I have a really, really big cat who might show up.
Eric?
Hello, I'm Eric. I'm a senior at the Bishop School, and my project category was bioinformatics. This is my second year doing science fair. Fun fact about me is that I really like cooking.
Yeah, if y'all haven't noticed already, bioinformatics is like probably the most popular topic at the science fair. So it's good that there's finally a new category this year just for us bioinformatics people. Sarah?
Hi, I'm Sarah. I'm a freshman at Canyon Crest Academy. My project category was biochemistry, and this is my second year in the science fair.
Badra?
Hi, my name is Padra. I'm a sophomore at the Bishop School, and my project category is also bioinformatics or computational biology. This will be my 4th year at science fair, and a fun fact about me is that I really love music.
Anirudh?
Hi, my name is Anirudh, and I'm a freshman at Mount Carmel High School. This is my second year doing science fair, and last year my project category was physics in engineering, energy, and transport. And fun fact about me is I like to play tennis.
Oh, me too. Okay, so that concludes our introductions. Thank you to all these volunteers for giving up their Saturday mornings to help out. So the agenda today will be, first of all, designing your statement of problem or purpose, such as like, how can we cure cancer? Or, well, that's very general. Or like, how can I improve the efficiency of a windmill? And then you're going to create your hypothesis, then the materials list, procedure, and then graphing your results. And finally, we'll be discussing where you can get online data for free if you aren't able to collect your own data. I know because with the COVID-19 pandemic, it's especially hard to, go into a lab if you want to do bio and like grow cells or whatever. So in some cases, like online available data is more feasible.
Hi everyone. So if you look inside of the GSDSEF website, under students resources, there is a sample notebook. And I remember when I was a freshman, this actually helped me a lot in writing the notebook because the process is a little bit more specific towards our fair. So our example is the effect of treatment of Raphanus sativa seeds on percent germination and growth rate. So these presentations are meant to help you write your notebook and cover the components in this sample. As a reminder, it's required for this year's fair, so be sure to take a look at that if you're new to the process. Science versus engineering and computer science projects. So a science project collects data and analyzes trends. An engineering project is a little bit different. You make prototypes and you don't necessarily generate the data. These projects are also accepted at our fair though. But an easier way to tell the difference between them is that the difference between building a rocket, which is engineering, and buying a rocket model and testing the different fuels for height is the science. If you have questions about what type of project you have, you can also ask in the breakout rooms later. Are we doing breakout rooms?
We will if there's enough interest at the end.
Okay. And then the first part of the notebook is the statement of problem. This is for science projects. It's basically a question or a statement that summarizes the problem you're investigating. And it gives both an idea of the problem and also it gives an idea of why it's important. So examples would be the effect of seed pre-treatment on radish growth, or which pre-treatment produces the best radish seed yield, which drug molecule will kill the most cancerous cells. So those are all ways you could write the statement of problem. It can be a sentence or a statement, either way. And then there's the hypothesis. This is also a really important part of your project notebook. In your project in general, this is for science projects. It's basically a proposed explanation or prediction about what you think will happen in your experiment. It's based on your research and the problem you've identified. It has to be testable and replicable, and it contains both the independent variable and the dependent variable. So you mentioned both of them inside of your hypothesis. And the format people usually use is if, and then insert the independent variable changes, then the dependent variable will be affected in some way.
So here are some examples for a good hypothesis. You can see the relationship between the if-then statement. So a higher concentration of aloe vera juice will decrease the regeneration time in planaria. So if higher concentration of aloe vera juice is given, then their regeneration time is decreased. And similarly, you can read the others, like if temperatures are raised in orange juice, then ascorbic acid concentration levels will decrease. Make sure if you haven't had a chance to think about your hypothesis, you should look at the independent and dependent variables of your project. And if you need help figuring this out, you can ask us later on. So one more back. So specifically for science projects, there's some things that you need to do. So you should be sure to state your hypothesis, and then you should design an experimental procedure. But in this, you should remember to include the control groups, ways that you can replicate your project, and then also ways that you're also randomizing your project. So there's an example of a control experiment or a control group in which there's one plant that is sprayed with the biofertilizer versus one plant that's not sprayed. So including these control groups are important. Okay, so if you're not doing a science project and instead you're prototyping and building or coding or engineering something, you can sub out the statement of problem and hypothesis with the statement of purpose and design objective. So this is basically why are you building and what are you building? So it's very similar to the statement of problem, but there's some slight changes in the wording. But again, if you're unsure of what type of project you have, since many projects seem to be in the middle of these two, you can also ask afterwards. These are also some examples of what an engineering and CS project would be. And some general notes for these engineering projects, you should always try to build a first prototype of software or your device, and then you can also include that building process of the prototype. That's something that judges like to see. And then you can see what's off and what's wrong, and then design a solution to help analyze these problems. Once you do that, you should build a second version and then again, see what needs to be improved. And then make sure you take careful logs of each step and each time so that you can show the progress that you made from start to end.
So now I'm going to talk a bit about the engineering design process and in a later slide, the scientific process. So for the engineering design process, you first need to define a need or express a goal. And in order to solve this goal or need, you have to establish the design criteria and constraints. And then you also have to think about alternative designs. And then you build a prototype of what you think is the best design. You test this prototype using the criteria, and then analyze the test results. You make design changes and retest. And then finally, you have to communicate the design. So basically just like explain what the design does and the results of the design. Okay, and this table is basically just comparing the scientific method and the engineering design process. So the scientific method is stating a question, doing background research, formulate a hypothesis and identify variables. And then you design your experiment, establish procedure, and then you test your hypothesis with this experiment. You analyze your results and draw conclusions, and then you communicate results. So it's kind of similar to the engineering design process, but there are some differences in which in the engineering design process, you kind of want to like test different prototypes and like go back, while in the scientific method, you really only want to like completely change your design procedure if it just doesn't work. And this is basically a really nice flowchart for both of these methods. That's pretty helpful in doing the experiment because, well, as you see, there's like a lot of different, there's a lot of good descriptions of what you do for these methods or steps. And it's also kind of useful in a lot of cases for like setting up a time frame or like setting up a schedule to do your experiment, because with these flowcharts, you can see like how much there is to do. And these are some really useful rubrics or guidelines that are on the GSD-SEF website. And for the so basically these, if you open them, they're just like a list of things that you want to do in these experiments. And if you just follow these lists in doing your project and get all of these conditions or like guidelines down, then your project is going to be much better.
Awesome. So next on this slide, we have a video comparing and contrasting science and engineering projects. So this could include the process, and it could help you determine if your project is science or engineering. And for the procedure, this would, for the materials, make sure to be concise with your materials so you can make a bulleted list of materials and exactly describe the amount and type of each material and piece of equipment. And make sure to only use metric system measurements. So like pounds or ounces, that would be the customary units. So you would want to use things like kilograms and grams, liters, milliliters, and that kind of stuff. So on pages 5 to 7 of the student notebook is your procedure. So you can put step by step, like 1, 2, 3, 4, 5, and etc. Or it can be in paragraph form. And we recommend you to use passive voice. So that an example would be 20 milliliters of acetic acid were added to the test tube, but using the active voice in science is also becoming more popular and accepted. For example, you could just write, I added 20 mLs of acetic acid to the test tube. But that's still acceptable, but we still recommend using passive voice.
Okay, so when you're writing up your procedure, it's really important that you list everything exactly how you did it. So for instance, if you used 10 to 250 milliliters plastic cups, make sure you also list the numbers very accurately. Numbers are really important when you're doing the procedure because it's supposed to be replicable. So you should use the exact numbers you used. So that's why in this procedure list, there's like a lot of numbers. Additionally, you want to have multiple trials. So for instance, at the end of steps 1, 2, and 3, there's like more trials. Important to get a lot of data. And then finally, you also want to use a lot of pictures when you're making your procedure to add a bit of like pizzazz, but also it's important to kind of show what you did. And then a bit more information on the procedure is you need enough trials to actually have like a sizable sample size. Trials are like how many times you repeat the experiment for every set of conditions. And then also you need sample size to be larger. It's like how many data points are in total or in like one trial. And then you should also have like a good number of both, depending on the type of experiment you're doing. And also what you like kind of read in the research should determine how many trials and everything you're doing, how big of a sample size and how many trials you should have is based on your research. Yeah, so you should just kind of base your research on like, or your procedure on the research.
Okay, so this is a really important reminder, but once you're finished with your with thinking about your procedure and engineering design, you need to submit your project to safety screening before you start experimentation. And the link below is like a really good resource that shows how to participate and like all the steps for this. And the deadline is really soon, it's before December. And you guys can ask us any safety screening specific questions if we do breakout rooms as well. But basically, everyone has to make an account and fill out a short safety form on the website and then submit it to the website. And if your project needs extra certification, you have to fill out additional forms. So for example, if you're doing a behavioral or social sciences project, you might need like a human consent form. Or if you're dealing with hazardous materials, you're going to need a form for that as well. And check, oh, sorry. We also have a YouTube series that explicitly goes through all the steps. That was
Sorry,
on.
I'm getting used to this like touchpad and mouse. That's why it keeps going back and forth.
Yeah. Yeah. The link on the slide, the YouTube link, goes to the How to Apply series on YouTube that shows how to do this. Yeah. Okay. So, when organizing results, after you collect your data, it's important to organize your data. So organizing it into categories definitely helps you interpret what's being observed. There are also some strategies when you're trying to organize your data into either tables or charts or graphs. Your independent and dependent variables are either going to be the rows or the columns in your chart or table or graph. And you should write down all your data because you can always put any raw data in your appendices even if you don't use it. And it's also good practice to write it in an Excel or Google Sheets table so that you can easily graph it. Graphing also is really useful because it illustrates relationships in your data that you might not have seen if you had just seen the numbers in a table or chart.
So making a table. So when you're making a table, there are two variables, the dependent variable and the independent variable. The independent variable goes on the left. So in this case, it's the number of weeks. And the dependent variable goes on the top. So it's, in this case, the total grass height. And the title of table goes on top. So that's the growth of grass. And the trials of the dependent variable are organized in subsequent columns. And it's important to do trials because you have to make sure that your results are consistent and not just by a random chance. And making a graph from a table. So this is the table that we have to make a graph from. So the dependent variable goes on the y-axis and the independent variable goes on the x-axis. So as you can see, the coordinates of the points are plotted on the graph on the right.
Okay, so now for the second part of the workshop, online sources of data. So I think that bioinformatics is going to be a very popular category, so I want to go over this really good resource. It's called the Gene Expression Omnibus, and it is sponsored by the government. And essentially, it's where researchers who do experiments about gene expression, they upload their data, and they have a lot of data sets that you can just use for free without even making an account. So if, for example, you wanted to study like lung cancer, you can just type in lung cancer here, and it's going to pull up a bunch of data sets. I can find an example one. Okay, so here's one. And you can download the data here, or you can use it here. I'll quickly just demo. But basically, this website allows you to find data sets with information and data from gene expression research studies, and also even analyze it all on their online platform. So it's really useful. There's not enough time in this workshop to explain how to actually do this stuff. But if you're interested, just Google like gene expression omnibus tutorial online, and there's a lot of materials. Yeah, pretty cool. So for example, this, just really quickly, this graph kind of shows the differences in expression levels of this RNA molecule between cancer and control. So you can see that this RNA is found more highly in cancer patients than in control patients. So that's just like a pretty cool analysis. Okay, so there's also the the TCGA data portal. TCGA is another thing by the government, and it's where lots of cancer research data has been published, especially on like gene mutations. So if we're going to do looking at like lung cancer, for instance, you can see like what are the common mutations that are occurring. Like KRAS is a very famous one. TP53 y'all might have heard of. So this is another super popular resource. And for instance, it's showing us that the most commonly mutated gene in lung cancer is TP53. So if you're more of an environmental person and want to look at climate data, the National Oceanic and Atmospheric Administration also has data that you can get to under this data access. Usually you often have to ask them for some more sensitive information, but for example, we could look at Arctic and sea ice, and there's so much data here, and you can like each of these links goes to a different data set. Ta-da. And yeah, so you can download it. Okay, so finally, there's also general data, and this is good for anyone to pay attention to if you're not doing bio and earth science, because this place just has so much data sets. So Kaggle is an online competition website where people submit data sets and enter competitions to see who can be the best at analyzing the data set, for instance. And there's just so much. Like if you wanted to do agriculture research, there's like images of plants that you can analyze, agriculture crop production in India, pesticide use in agriculture. There's even music, Spotify music analysis, music features, Amazon musical instruments reviews, music genre classification. And of course, there's like cancer stuff, history stuff, there's even history stuff. So Kaggle, Kaggle.com is a really great website. And it also lets you program in your browsers, so you don't have to download stuff. I don't have an account signed in right now, but you can see like in this data set, people have public code that you can take a look at and kind of learn that way. So it's a really great resource. So this one, for example, is kind of mapping, plotting the different genres of music into a graph based on their characteristics, I assume. Okay, so thank you so much for coming. I will be emailing, I don't know if I can share the slides, but I will be emailing the important links that were shared in this presentation, such as the YouTube videos, the Kaggle, all these. Thank you, Kavya, for putting the workshop in the chat. And the next workshop is December 11th, so quite a while away, and it's on data analysis and statistics, and we'll be teaching how to do that using Google Sheets. And make sure to tell your friends. If you have any questions, email gsdsef@slb@gmail.com. And you can register for the next workshop sent by Kavya. It's the same form as the one you used to sign up for this one. So if you've already filled it out, no need to fill it out again. So I'm not sure how many people are still here, but if you are interested in like a breakout session right now, just private message me, please, so I can get a measure of interest. And if not, no worries. Yes, the workshops will all be uploaded on YouTube. So it'll be out maybe 3 to 4 days after the webinar happens, just to allow time for our video production committee to upload it and process it. Okay, so it looks like we have 2 students who would like breakout rooms, and we're a little bit ahead of schedule, so that works great. Just 1 second. Okay, so if you would like help, please go to breakout room 1 or 2. If not, thank you so much for coming, and I hope to see you at the next workshop.